for potentiometry in your syllabus, what we are need to emphasize is principle, working, then ion selective electrodes and applications of potentiometry. So we will focus on that part only. There are many sub points. There are many things to be studied in potentiometry, but what I intend to do is we will focus on the things which are there in the syllabus only. Other things we will just skip from the slides. Okay. The principle behind potentiometry, the principle involved in potentiometry is when pair of electrodes are placed in sample solution, it shows potential difference by addition of the titrant or by change in the concentration of the ion. So in simple terms, suppose there is one solution in which there are ions present. You measure its potential. That solution ka potential you need to measure. Now, when we say potential, it has to be with reference to some other solution. So take a fixed standard solution. Against that solution, you measure its potential. If you change the concentration of your solution or you change the composition of the ions present in the solution, the potential associated with the solution will change. As simple as that. Okay. So the original potentiometric methods, one schematic diagram is given over here. There are two different solutions. In two solutions, two electrodes are kept. These two solutions are connected via a bridge. It is called as salt bridge. This bridge allows the contact between two solutions, and through this bridge only ions can pass through. The solvent and other contents will not be able to pass through. Okay. And the potential difference between the two solutions is measured over here by a galvanometer. One of the electrode in which there is no sample present, a standard is present, is called as the reference electrode. The part of the cell that is held constant, so composition of this solution will be held constant. And then there is indicator electrode, the part of the cell that contains the solution we are interested in measuring. So analyte will go here. Here there will be standard. The potential difference between two electrodes will be measured. So what is the reference electrode? A reference electrode is the electrode which contains of its own potential value and it is stable when dipped into sample solution. The salt bridge is used to prevent the interference of the analyte solution with that of the reference solution. Now, here when we say analyte solution, the analyte solution is a solution whose potential is to be measured. Okay, it may be the titrant of any titration, or it may be your analyte whose concentration you want to measure. So any solution, analyte solution. Okay. Now, the indicator electrode is the electrode which responds to the change in the potential of the analyte solution. So in simple word, the electrode that is dipped in the analyte solution will be called as indicator electrode. The electromotive force, EMF, okay, of the complete cell, this will form the complete cell, entire assembly. So electromotive force of the complete cell is given by the following equation. EMF of the cell is equal to EMF of the reference electrode plus EMF of the indicator electrode plus EMF at the junction. Okay, now what is this E junction? E junction is electromotive force of the reference electrode. Okay, E indicator is electromotive force of the indicator electrode. E junction is the electromotive force at the junction of the liquid, that is the salt bridge. Now, the theory behind this method. The main theory involved in potentiometry is when known potential electrode. Okay, now listen to the words carefully. Known potential electrode, the electrode with known potentials immersed in the sample solution when the potential is given by Nernst equation. Nernst equation is given here. E, that is potential of the solution, is equal to E0. E0 means it is the can say potential of the standard electrode. Okay, standard electrode potential plus 0.592 divided by n entire thing multiplied by log C. Now, n is velocity of the ions, C is the concentration of the sample solution, and 0.592 is RT divided by F. Okay, so actually it is see, if you will try to simplify it, it will be RT divided by NF. But RTF are constant, its value becomes 0.592. Now, where R in RT divided by F, R is the gas constant, T is the absolute temperature, and F is Faraday's constant. Now let's go a little more in the theory part. What are electrodes? These are mainly used to measure the voltages. Mainly two electrodes are used in potentiometry, namely a reference electrode and indicator electrode. The reference electrodes are mainly used for the determination of the analyte by maintaining fixed potential. Example of reference electrode is standard hydrogen electrode, silver chloride electrode, and saturated calomel electrode. Now don't worry, you don't have to study these electrodes in detail right now, okay? Not in your syllabus, so just remember their names, that will be sufficient. The reference electrodes are classified into two main classes are as follows. Primary standard electrodes, the example is standard hydrogen electrode. Okay, Mr. Shubham Nikam has joined. Okay, where were we? Okay, classification of the reference electrode. It is primary standard electrode and then comes secondary standard electrodes. Now, as the case of standard substances, primary and secondary standard, there are differences between the primary standard electrode and secondary standard electrode also. Now, can anybody tell me if you can remember what is primary standard electrode and secondary standard electrode? Does anybody remember? शायद ऐसा लगता है किसी को भी याद नहीं है. See, primary standard electrode, you remember that it has got zero potential, okay? And for secondary standard, it has got a fixed standard, fixed potential. But for this electrode, the potential is always zero, okay? So next type of electrodes are indicator electrodes. We are done with primary electrodes. Now comes the indicator electrodes. It is used to measure the potential of the analyte solution comparing with that of the reference electrode. Its potential is directly proportional to ion concentration. If ion concentration changes, the potential at the indicator electrode will also change. Examples are hydrogen electrode, glass electrode, antimony oxide electrode. You must be very much familiar with the glass electrode. All those pH meter experiments you performed, those were glass electrodes. Now, there are two classes of indicator electrodes. First is metal indicator electrodes and second is ion selective electrodes. Now, these ion selective electrodes are there in your syllabus. We will quickly see what are these metal indicator electrodes. We will not go deep into that, just we will see their definition. These metal indicator electrodes develop electric potential in presence of a redox reaction on the metal surface. Platinum or silver are used as metal indicator electrodes. These are mainly classified into three types. Let's now go into further classification of that. Okay, we are interested in these ion selective indicators. Now, when we say ion selective indicators, does that word strike something in your mind? It is electrode, so it is basically it is related to potential. When we say ion selective, what does that strike in your mind? Yes, Shakil, very good. So it will detect only one ion. Instead of saying that, let us say they will be responsive against or responsive for only certain type of ions. If the concentration of some other ion changes, they will not give such type of response. Okay, the excellent example is glass membrane electrode that you were using for measuring pH. Okay, so this is that glass electrode. The other example is antimony oxide electrode. Okay, just first observe the construction of this glass electrode. I will enlarge the image. अच्छा, हमने definition देखा ही नहीं officially. I have not read out those two lines. So these ion selective indicators are composed of ion selective membrane by which the ion crosses and it creates imbalance. That means it creates the potential difference. Observe this diagram on your right. Let us start from the bottom. There is sensing bulb. It is in a way you can say a membrane, glass membrane, which allows passage of ions. Then there is ceramic junction between the glass tube and this sensing bulb. Now, in this glass tube, you will observe different arrangement. There is one spiraling wire that is coming down. It is your reference. Okay, so it is a reference wire. Okay, at the center you have a tube in which one wire is suspended. Okay, this is the silver wires dipped into AgCl solution. So this is internal filling solution. So there is a tube inside a tube where AgCl solution is present. Then there is one outer fitting, and to refill the solution you have a fill hole over here. Then there is next type of electrode, antimony oxide electrode. Observe this diagram now. Okay, basically this entire assembly will look like a closed cuvette, small box that is provided with these three wires. One is a reference electrode, the other is working electrode, and the other is counter electrode. Okay, so there are three electrodes. One electrode, working electrode, is dipped inside the solution. Okay, the liquid is filled till this level. Then there is a reference electrode and counter electrode. Now this much diagram, and if you remember, that is sufficient for you. These are types of potentiometric titration. Let us not go into that, not in your syllabus. Then there is acid-base reaction, precipitation reaction titration, redox titration, complexometric titration. Then the method, how the experiment used to be done. You people have done these experiments actually. So let's directly jump on to applications of potentiometry. So first application is in clinical chemistry. Ion selective electrodes are present sensors for clinical samples because of their selectivity for analyte in the complex matrices. So in biological sample there are many ions are present. So certain type of ions can be only detected by the potentiometry. The most common analytes are electrolytes such as sodium, potassium, calcium, hydrogen, chloride, and dissolved gases such as carbon dioxide. Then applications in environmental chemistry for analysis of cyanide ion, ammonia, NO3, F3 in water, and also in the analysis of wastewater. Then comes potentiometric titration in the lab for determination of equivalence point of an acid-base titration. It is also possible for redox, precipitation, acid-base complexometric titration, as well as for non-aqueous and aqueous solvents. Okay, then application in agriculture, determination of NO2, ammonia, iodine, calcium, potassium, cyanide, chlorine in soil, plant materials, even feedstuff in the animal industry. Then for fertilizers, and applications in detergent manufacturing, determination of calcium, barium, fluorine, and effect of these on water. Then next is application in food industry. In food industry, lot of ions can be determined using this potentiometer. So salt content of meat, fish, dairy product, fruit juices, brewing solutions, calcium in dairy products, also in beer, potassium in fruit juices, in wine making. Then corrosive effects of NO3 in canned foods. So NO3 is many times produced inside it. Then fluorine in drinking water and other drinks, NO3 and NO2 in meat preservatives. Then assay of bisacodyl suppositories and assay of sulfa drugs can also be determined by this method. Now with this we are done with our entire syllabus. I will share these PPTs on our group also, on Google Classroom also.